Hello, everyone. Thanks for your interest in my research. Here, I talk about conserved lysine residues of HBV core protein are not required for cccDNA formation. There are two lysine residues on the HBV core protein, K7 and K96, which are located on the HBC protein surface. These two residues are highly conserved across all also hepatinovirus core proteins. These two lysine residues have been proposed to function at various stages of the HBV life cycle, potentially through the lysine-specific post-translational modifications. The goal of this study was to investigate the roles of the lysine residues on core protein in the HBV life cycle. So we made the mutations on either K7 or K96 or both residues to the alanine or arginine to investigate their effects. We made the mutations in the context of the HBV full-length replicon or the HBC expression construct. In this case, the HBC expression construct was co-transfected with the HBV C- replicon. After transfection into the human hepatoma cells, we find all these mutants could support the HBC expression, capsid assembly, and pgRNA packaging, suggesting the HBC K7 and K96 were not essential for capsid assembly or pgRNA packaging. Next, we looked at the effects on the reverse transcription. We find none of these mutants showed the defects on the DNA synthesis, suggesting the K7 and K96 were not essential for HBV reverse transcription. We also investigated the effects of the mutations on the viral secretion. We find K96A or K96R or double mutant showed severe defects on the HBV viral secretion, but the HBC K7A, K7R mutant showed the similar viral secretion levels as a wild type. This suggesting the HBC K7 was indispensable for HBV viral secretion. Next, we investigated the effects of these mutations on cccDNA formation. We find all of these mutants could support the cccDNA formation after the transfection in the human hepatoma cells. And we normalize the cccDNA levels to the rcDNA as an indicator of the cccDNA formation efficiency. We find HBC K7A, K7R showed similar levels on the cccDNA formation as a wild type. Interestingly, the K96A, K96R showed about 3 to 6-fold increase on the cccDNA formation compared to the wild type. This result clearly suggests the K7 and the K96 are not required for the cccDNA formation via the intracellular amplification pathway. On the other hand, the nucleocapsid uncoating and the rcDNA rcDNA nucleo import, which are two prerequisites for the cccDNA formation. Our results clearly suggest the lysine residues or the post-translational modifications on these two lysine residues are not required for this two process. As the K7A and K7R mutant remain able to support the viral secretion, we investigated the effects of the K7 residues during the de novo infection. We used the inoculum made from the full-length replicon transfected cells to infect the human hepatitis 2 NTCP cells. We find this mutant still can support the cccDNA formation, but these mutant infected cells showed severe defects on the RNA production. This tells us the protein K7 mutation is not required for the cccDNA formation, but this mutation has both the defects on the mutations on the core protein and the genome has the defects on the RNA production. So next, we made another inoculum which has the wild type C- genome, but has only the mutations on the capsid protein. We find they can support the cccDNA formation and also could support the RNA transcription. This tells us the HBC K7 was not essential for the HBV transcription from the cccDNA. This may indicate that the genome of the K7A and the K7R mutation maybe has some cis defects. To further test that, we made another type of the inoculum, which uses the wild type capsid, but the mutant genome, which genome carries the K7A or K7R in the context of the wild type C- replicon. So we find this mutant can support the cccDNA formation, but they could not support the RNA transcription. This suggests the coding sequence of the HBC K7 was indispensable for the RNA production. When we looked at the coding sequence, we find is AAA, which is a part of the HBV polyadenylation signal UAU AAA, which is indispensable for the RNA production. Altogether, our result has indicated the HBC K7 and K96 were dispensable for all intracellular replication steps. And the K7 and the K96 or post-translational on these two residues were not required for the nucleocapsid uncoating, rcDNA import, and the cccDNA formation. And our results also indicate the HBC K7 or post-translational modifications on the K7 were dispensable for the de novo infection. Instead, the coding sequence of HBC K7 AAA is a part of the poly-A signal UAU AAA, which is critical for the viral RNA production during the infection. At the end, I would like to thank for your attention, and I'm happy to take any of the questions. Thank you.